Hello! Right, here I am back for another video. Right, so today I'm going to talk about flow in a reef tank, and I'm not going to talk about pumps and how to put flow in your reef tank. There's lots of videos out there which tell you how to set up flow in a tank, why it's important. So there's no point repeating that, really, is there? So I thought I would just go down a level rather than talk about how to initiate the flow in a tank. I thought I'd talk about why that flow is important in terms of the biology of the coral and what's going on with the coral and the flow. So essentially I'm going to be talking about SPS coral. I guess it is applicable to LPS and soft corals, but essentially I'm going to talk about SPS coral. So where should I start? So flow. I guess if I start with the biology of the coral itself, and then we can work our way up to the flow side of things. So firstly, if we think of SPS coral, they're quite unique in the fact that they have all four trophic levels of feeding available. They can carry out all four, and so trophic levels of feeding are basically primary producers, secondary and tertiary consumers. So a primary producer is something which can take inorganic molecules, reform them basically and make organic molecules and build biomass from that, which is what plants do, and it's that process called photosynthesis, which we all learn at school, where we take light, carbon dioxide and water and make complex molecules from them. So corals, we know, can do that. They can also be primary consumers in the fact that they can take those compounds which are made in photosynthesis and use them in their tissues to build and grow and build biomass, basically. And then they are also able to be secondary and tertiary consumers in the fact that they can trap and feed on and get nutrient from phytoplankton, zooplankton and bacteria, for example, which they can take through their tissues by capturing on their tentacles with their nematocysts, those stinging cells, and bring them in. And it gives them things like nitrogen and phosphorus, which are not available through the photosynthetic pathway. So in that respect, they're quite unique in the fact that they've got all of those trophic levels available to them. Why is that important? I guess, one example would be, so let's say you black out your tank, so you have no light. So we know we keep going, corals are photosynthetic, so if we give them no light for 4 days, they're all going to be dead, yeah? Well, they're not, are they? So if you treat your tank for cyanobacteria or dinos, and you black your tank out for 4 days, most of the time, if you're lucky, when you take the covers back off your tank, your corals are going to be still alive. They're not going to be happy, they're going to be still alive. Now, why is that? Is they're photosynthetic? Well, the answer there is, one, they have reserves, like most people or most animals do. They have a certain reserve for times when nutrients are scarce. So they have they have lipids available to them, carbohydrates available in their tissue that they can use to see them through those times when photosynthesis is either low or not being carried out at all. And then, as I already said, they're secondary and tertiary consumers, so they are able to capture particulate matter, phytoplankton, bacteria, zooplankton, which will give them certain nutrients which they need. So for the short term, and it differs between species of coral, some can go a number of days without the light and photosynthesis, some can go for weeks. I think things like gonipora, jumping out the SPS thing, they can go for even into months actually without the light. And it's just like the kind of percentage, I guess, of secondary and tertiary consumers which they use to get their nutrients. Whereas SPS corals, it's generally thought that 80 to 90% of the nutritional needs are met by those photosynthesizing dinoflagellates which live inside their tissue. So anyway, so that's the kind of basic biology of the coral, the trophic levels that are available to them, and why they don't die instantly when there's no light. So if we go back to the actual structure of the SPS coral itself, so if you look at that diagram there, it just shows it's a very simple structure. You've got an epithelial layer, an endothelial layer, a sac-like structure called a coelentron, and there's also a gastric vascular canal, which polyps in this colonial organism can share nutrients basically. And that's why it's called a colonial animal and not just a lot of single animals living on a close space next to each other. And they do actually interact with each other, which makes them colonial. So within that tissue of the coral, there is a, I want to call it like an organelle, I guess, and it's called a symbiosome, and in that is the dinoflagellate, which is responsible for the photosynthesis. So it's got these little chloroplasts in there, which has the chlorophyll, the pigment, which is able to convert the sunlight and use it to make inorganic molecules into organic molecules. So you need light, carbon dioxide and water, and I'm not going to go through the process of photosynthesis, but at the other end you'll get things like glycerol, glucose, essential amino acids and lipids, and then they get translocated from that area into the rest of the tissue of the coral, and they allow it to metabolize and grow and produce gametes and do all the things that an animal does. So why is flowering important? Well, as you've already said, because once that planular larva sits on a rock and starts to grow and turn into a coral, it has no mechanism to go and get its food. So it's there, it's stuck there, and the only way it's going to survive is if it gets washed with nutrients which are available to it to bioassimilate, to grow basically. And so the only way it gets out is through current. So everyone knows that really, who's got a reef tank anyway. But on a more kind of finer scale, what we have is boundary layers which surround the corals. So there's three essential boundary layers. So if you look there, you can see the three layers that I'm talking about. So you have a benthic boundary layer, a momentary boundary layer, and a diffusion boundary layer. So I think in terms of the reef tank, benthic boundary layers not really relevant, I don't think. Benthic just means on the bottom basically. But momentum boundary layers we certainly have in our reef tanks, because that is basically just the disruption of the water when it hits our branching corals or our monty caps or anything. It disrupts the flow of water in our tank. So if you think of all the branches of your coral, and as the water hits it, they're going to be going round, under, over, and that disruption of the current is the momentum boundary layer. So it's quite close to the branches of the coral, but obviously the water either side and above it, which is unimpeded, just goes past at a faster rate. So a boundary layer is basically caused when the water hits the surface and is slowed, and the water above it is unimpeded and travels faster. So you get a laminar kind of effect. So you get a slower layer of water underneath a faster layer of water. And that's basically a boundary layer really. So in terms of our coral, I've already said we get a momentary boundary layer, and then beneath that we get a much thinner diffusion boundary layer. And that's where all the kind of essential nutrients that a coral needs are pulled out of the water column and into the tissue of the coral. And it's like a diffusion gradient really. So that transport across the membrane can be either passive or active. So passive means there's no energy involved in that process. So if you look at, if you just look at like osmosis, for example, as a classic, not a passive way to get molecules across a membrane. So there's no energy involved in that. But other things do involve energy, and you are selecting specific molecules and drawing them in, and there's an energetic cost to that. So all this takes place because this endosymbiont, this dinoflagellate, sits within the tissue of the coral, and if it doesn't get what it needs, obviously it's going to die because it's trapped in the tissue. So the coral has to bring everything across this little boundary layer and into the tissue in order for the dinoflagellate to live, and also for the coral to live, because once that photosynthetic pathway produces those elements I mentioned earlier, that's what the coral tissue needs to grow, survive, produce gametes. So what is a boundary layer? There's two kind of ways to look at it. One is a definition of a boundary layer. So basically the scientific definition is the boundary layer is the layer in which the solutes in that boundary layer are greater than 10% plus or minus the ambient salt water outside of it. So when you get to that 10% plus or minus whatever you're looking at in the boundary layer to the ambient salt water, that's the kind of edge of the diffusion boundary layer. In terms of actual size, like measuring it, it has been done. So they looked at two corals, one was Parietes and one was Montipora. And it was actually thicker than I thought, because I thought it'd be measured in like microns. I expected it to be like super thin, but actually it's a lot thicker than I thought. So in Parietes they measured it, this is in artificial conditions in a lab, not on a reef. But they measured it in Parietes, it was 1.5 millimeters, I think it was 1.53 to be precise. And then on the Montipora it was just over 2 millimeters, 2.06. Plus or minus half a mil, I think. So it was quite thicker than I thought, to be honest. So why is it important? Well, it's just because that is the essential part that all the nutrients that the coral needs have to come across. So it just pulls them out of that very thin layer. So if that thin layer is not constantly replenished, then you can have all the stuff in your tank you need. But if you haven't got the flow to get it close enough to the coral that it can pull it out, because the coral's not going to be able to pull out, you know, if it's got bicarbonate in the water but it's a foot away, it might as not be there, because you can't get it, you can't access it. It's got to be right up next to, within a millimetre of that coral tissue in order for the coral to be able to utilise it. And flow has an inverse relationship with this diffusion boundary layer. So basically, you increase your flow, you'll decrease your boundary layer. If you decrease your flow, you'll increase your boundary layer. So there's that inverse relationship. And that's why we talk about having dynamic flow. So you want basically to keep washing that boundary layer with fresh water. So it's going to do two things basically. It's going to bring all of those nutrients that coral needs, all those inorganic carbons, and it's also going to be flushing away all of those waste excretory products which have to go through the tissue. So the only way that coral communicates with the outside world is by diffusion, really. Which is why they grow the way they do. Which is why most people think they're plants. You know, outside of hobbyists, if you haven't got a biological kind of background, zoology or whatever, you look at a coral and you think it's a plant because they look like plants in the way that they grow, and they act like plants really. And that's because essentially the zooxanthellae in the tissue is like the chloroplast in the leaf of a tree or a plant. And they grow in similar, you know, branching coral, when you look at it, is not unlike a tree, in the branches of a tree. And it's trying to maximise, one, the surface area, which is in contact with the water around it, and then it can diffuse across that vast area of tissue. And two, it wants to get as much light onto those cells in that tissue as possible, which is essentially what a plant's doing, really. So that's why they look rather similar, but obviously there are essential differences. As I said, they have all four trophic levels available to them, which plants don't, for example. Yeah, so the boundary layer is important, the flow is important, and so what are the consequences of low flow? So if we look at flow in terms of linear speed of water, which is how it's measured in all of the scientific papers that look at these things, and unfortunately you can't relate that to our reef tanks, because as far as I'm aware, if someone can correct me, then please put it in the comments. I couldn't find any kind of linear measurements of flow for pumps that we use in our tank. So they all use gallons per hour, litres per hour, like how much water they move. But they don't say like 50% an MP40 will move 10 centimetres a second, for example. And that's how, unfortunately, all journals, all science measures flow. So slow flow we're going to say is 2 centimetres a second and below. Moderate flow is 3 centimetres to 7 centimetres, and high flow is 7 centimetres and above. And they can go up to 70 centimetres a second, even a metre a second in super high flow. So, excuse me, I'm just having a dry throat. Yeah, so the repercussions of slow flow. So if you you want to skimp on your flow in your tank, or you're transitioning from soft corals up into SPS and you're not sure how much flow you need, the consequences of low flow, as I've already said, there's an inverse relationship. So low flow, you're going to have a thicker boundary layer, and that means that water's not being replenished as much as it is in the higher flow. So if it's not replenished enough, they've measured it in low flow. I can't remember what coral it was, but basically they saw a 25% reduction in the photosynthetic rate of the coral. So it's reduced by a quarter basically. So in daylight hours, that coral is going to be growing and producing nutrients 25% less than if it had good flow. So why is that? Well, that's because the boundary layer is going to be thicker, and it's also going to be replaced slower. So that means that coral is pulling out that inorganic carbon that it wants, but it's not being replaced quick enough, and the excretory products which are going into that boundary layer are not being removed quick enough. And so those two factors reduce the rate that photosynthesis can take place. And so all those metabolic pathways are slower than if that flow was greater. So essentially, I think that kind of explains why we want flow in our tank. Obviously there's an upper limit to your flow as well. So you don't want to be blasting your corals, because if you put strong enough flow on a coral, you won't have a boundary layer at all, and what you'll actually do is you'll rip the tissue off your coral. So the boundary layer will be non-existent, and all you're doing is actually putting the cellular structure of the coral apart. So you don't want that, obviously. But there's quite a large range in between. But really, you don't want a flow which gives you too big of a boundary layer. So I think that kind of, I hope that explains kind of what a boundary layer is and why flow is important. It's just a little bit deeper than going, you need flow to bring nutrients to your coral. So I just thought, I found it quite interesting, so hopefully some of you might find it interesting. And essentially that's it. So I can't really see any point of going deeper. I mean, if I'm honest, most people don't watch these videos anyway. I find, I'm a scientist, aren't I? So I'm going to find it interesting. You know, I do a lot of reading. I'm just kind of moving over from, I did a hell of a lot of reading about fish when I was setting the tank up, because I wanted to quarantine all my fish, and I wanted to know the fish biology, I wanted to know all the parasite biology. So now I'm kind of done with the fish in the tank. I'm moving on to the corals, and I just like a kind of a depth of knowledge. And that's just me personally. You don't need to know any of this. None of this is relevant. There's loads of people out there with absolutely stunning tanks, you know, packed full of SPS coral, and they know they need flow, and they provide the flow, and they have wonderful coral, and that's the end of it, and that's fine, and that's perfect. But there's a small percentage of people like myself, I guess, who like to know why it's important, and I like to know exactly how that coral's getting its nutrients, what it does with those nutrients, and I just find it interesting. So that's just the way I am. So hopefully there might be a couple of other people out there who also think like that. But yeah, the reality is most people don't watch these videos because I guess they're just not interested, which is fair enough. You know, that's what I like about this hobby. Actually, this is a really good hobby in terms of, you know, you can go in as shallow or as deep as you like in all facets of this hobby, really. So for someone like me, I like to dive deep, and it's good. You know, it keeps me occupied. It gives me an interest, and it keeps me kind of mentally a bit sharper than I would if I sat there watching shit telly, basically. So yeah, so that's it. So right, so I hope someone found it interesting. My next video is just going to be my weekday maintenance update, and yeah, so that's it. So I will see you in the next video.